I spent some of last class talking about the concept of linkage and hitchhiking and how sometimes it's bad for hitchhiking to occur when, what's the concept? What happens in hitchhiking? How many mutations are involved? At least 2, okay. What do we know about those mutations if hitchhiking is occurring? One's good, one's not. Very good. And what's the net, if you added up how good the good one is and how bad the bad one is, what's the net effect? The overall, is it overall good or overall bad to have the combination of the good and the bad mutations? So we talked about Crohn's disease and the idea that there's a beneficial mutation in this gene called OCTN1, otherwise known as SLC, whatever that arrow is pointing to, that increases dietary antioxidant uptake, and it happens to be located on the exact same piece of DNA, the same chromosome, as a mutation in IRF1 that seems to cause, or at least be related to, Crohn's disease. So we get a beneficial mutation and a detrimental or negative mutation. And how far apart are these on the chromosome? One's at 131,600,000 base pairs on chromosome, what chromosome is this? 5 in humans. The other one's at 131,800,000. So they're 200,000 base pairs apart. These 2 mutations found on the same chromosome. So this is a haplotype, 2 linked mutations. And they travel together from individual to individual, from parent to child to grandchild, grandchild to great-grandchild. And these scientists, we talked about last time, performed a study where they looked at the amount of gene expression occurring from each of the genes in this region of chromosome 5, where this disease had been genetically mapped. And today, the concept that we're talking about that's new is genetic mapping. So we're going to do this a little bit backwards. This is the outcome of genetic mapping. Scientists identified this tiny little region of chromosome 5 that they thought had a mutation that caused Crohn's disease. And they found a single gene, IRF1, where there was a significant difference in how much of the gene was produced in individuals that had Crohn's disease versus individuals that did not. And so that suggested to them that it was this mutation in IRF1 that's correlated, at least, with Crohn's disease. So we have a bad mutation there, presumably, and a beneficial mutation in OCTN1. 200,000 base pairs apart on the same chromosome. This is where recombination is supposed to come in and save the day. We've got a good mutation, we've got a bad mutation, and they're physically linked. So what would be useful? What would help the situation? So up top here we have the version of chromosome 5 that has a bad mutation in IRF1 and a beneficial mutation in OCTN1. Here we've got a second chromosome. It has no mutations. It neither has the good one or the bad one. So imagine that this is 2 gametes. There's a chromosome 5 in one gamete. I'm going to use the Roman numeral just to distinguish it from other things. So let's say that's an oocyte and this is a spermatid. Flagella, chromosome 5. If those 2 fuse and you get an individual that's got one of these sorts of chromosome 5s, and a chromosome 5 from dad transmitted by the sperm that has no mutations at all, what's going to happen to this individual? Will they have Crohn's disease? What do you think? Got the bad mutation, the red one. They get Crohn's disease? If you don't want to answer, then ask me a question.
Is mutation is a gene response for Crohn's disease recessive or?
Fabulous. Yeah, so if you didn't hear in the back, the question is, okay, so what we need to know is the red mutation dominant or recessive? So let's assume it's dominant. Would this individual have Crohn's disease? Yes. Except I don't know how to spell Crohn's disease. Okay, would this individual, let's assume that the blue mutation is dominant as well. Does this individual have increased dietary antioxidant uptake? Yes. Is there a way to get that beneficial blue allele on a different piece of DNA than the red mutation? To separate them so that this individual could have the opportunity to have offspring that only get the beneficial allele, not the red, the detrimental allele. What sort of process would do that? Would separate these two mutations? Recombination. So after DNA synthesis, if this was a cell that was undergoing meiosis, post-synthesis you'd have a tetrad. You have one copy of the chromosome that has the two mutations, the good and the bad together, and you've got two wild type that were inherited from the dad. So if recombination happens between those two mutations, then this individual winds up producing 4 types of gametes. So I'll just letter them or label them A, B, C, and D. So those chromosomes then get segregated into 4 products of meiosis. A, nothing happened, no recombination, so you get the blue mutation and the red mutation happening on the same chromosome, transmitted together. D is fine. It never had the mutations to start with, so we're going to give it a star. And it still doesn't have either of the mutations. What happens in B and C? So here's chromosome B. It gets 1 red mutation. And then chromosome C gets 1 blue mutation. Like this. Crossover. So recombination has separated the two mutations from each other. So this individual, because recombination happened between these mutations, this individual that's producing these eggs or sperm has the opportunity to pass on a gamete that carries just the beneficial mutation by itself, without that tag-along, hitchhiking, deleterious mutation. So how do we know whether or not this can happen and what rate this kind of recombination can happen at? What do we need to know? Sort of generically, qualitatively. What do you want to know about those two mutations to know how likely is it that a crossover is going to happen between them and give us the ability to produce offspring that just get the beneficial allele? What dictates the rate of recombination between two genes? Yeah, physical distance between them. So how close are these two mutations? If they're really far apart on the same chromosome, then recombination is likely to happen between them. If they're really close together, not. So that's what's drawn here on this next slide in this set. So you got the 2 linked mutations. And many, many years ago, if we look at just a sample of 5 different versions of chromosome 5 from the human population back in time, if we hopped into our time machines, you might see that there are a couple of different haplotypes. There's one that just has the bad mutation, the red mutation. There's one that's got the good and the bad together. And then there are a bunch of chromosomes that have neither. So these are chromosomes that you might have taken from different individuals at some point in the time long ago in the past. Which one of these chromosomes is most likely to be passed on to the next generation successfully? So over generations and generations of time, if you revisit this population later, which one of these chromosomes is most likely to still be present? A, B, C, D, or E? Which one's got the most beneficial combination of alleles overall? Which one hitchhikes? A. Because we said earlier there's a net benefit. Despite one mutation being bad, the overall benefit of the beneficial allele makes that chromosome survive, this top chromosome. What happens to the second chromosome, the one with just the red mutation on it? What happens to individuals that inherit it? Are they healthier or less healthy? They're less healthy, right? So if you come back to this population way in the future, if you sampled 5 chromosomes out of the population, what would they look like? Would they mostly look like the top chromosome, the second chromosome? Say 1,000 generations later. What do you think? Is there a benefit or a cost to inheriting chromosomes C, D, or E up here? What happens to you if you inherit one of those chromosomes? They're not good, they're not bad. They don't have either of the beneficial or the detrimental mutations. So we'd call those neutral chromosomes. They don't do anything useful for us, they're not detrimental to us. So if you come back years later, you expect to see some of these chromosomes still in the population. They haven't been selected against. The individuals that have those chromosomes haven't been naturally selected, removed from the population. Okay. So when we come back a few generations later, if there's a net benefit to that combination of the black and the red mutations, the beneficial and the detrimental mutations, you come back a few more generations, you might see that that chromosome that has both of those mutations together is in more individuals in the population. The first individual that had that combination of mutations had kids that happened to have more kids who happened to have more kids that passed that beneficial combination of mutations on to their future generations. What's going to happen to the just black chromosomes, the ones with neither mutation, over time? We're starting to see there are fewer of them now. What happens eventually? They totally disappear, you're right. So that's what happened to the human population, essentially, not entirely, but almost essentially now. That combination of good plus bad mutations has swept, what we call sweeping through the population. They moved rapidly through the population of organisms, humans in this case, so that now almost everybody has this combination of mutations. And there's no way that we can get back the original chromosome or an original chromosome that just has the detrimental mutation, which is good because we wouldn't want that. There is also no way to get back a chromosome that just has the beneficial mutation unless we can happen to have recombination by chance land in between those two mutations and separate them like we drew earlier. Oh, no, that's okay. So this is the point I'm making. We know that those two mutations are about 100,000 base pairs apart on chromosome 5. Chromosome 5 is 180 million base pairs long. So if you wanted to, you could come up with a simple test to see, a simple statistical calculation. What's the probability, if recombination can happen equally likely at any point along the chromosome, what's the probability that recombination occurs between these two linked alleles? You just divide them. So if you've got 180 million base pairs of chromosome and you're looking for a recombination to land in a 100,000 base pair interval, what percent of the chromosome is that? It's like 1/1800th of the chromosome. So the probability that we can recover the beneficial mutation on a chromosome by itself without that hitchhiking bad allele tagging along is about 1 in 1,800. This is not very likely. So those two mutations move through the population hitchhiking. Last questions or concerns about hitchhiking? Concept might show up on the exam. Might. We've got lots of time next class to discuss this more if you want to take some time and reflect on it. Or if you don't, your choice. So if not, then we move on to some new topics, not for the exam. Do still pay attention, of course, please. But this is related to what we've just been talking about. So genetic mapping. How do geneticists figure out what parts of what chromosomes contain genes that control traits like diseases? So in other words, at the start of the Crohn's disease story, what did geneticists do potentially to figure out that that part of chromosome 5 contained a gene that controlled Crohn's disease? I won't be talking about Crohn's disease anymore. We're going to switch to a couple of different conceptual examples of how genetic mapping works. We probably won't have time to hit quantitative trait locus mapping today. We'll see. So here's the concept of genetic mapping. So imagine that you've got a trait like skin color. And let's assume in this case, whatever organism we're working in, that skin color is controlled by a mutation in a single gene. And that there are two alleles. So we've got big A and little a. And what I mean by discrete trait values is that there's either light skin or there's dark skin. There's no intermediate. Except if you're heterozygote. So there are three different values of this trait. There's light, there's medium, and there's dark skin. So what does it take to get dark skin in this example? What genotype do you have to be? So you have to be the darkest possible of these three colors. So each of the boxes represents the phenotype. We've got light skin, we've got dark skin, medium skin color. What genotype do you have to have the darkest skin color? Homozygous big A. Okay, so every time we see an individual up here that has two big A alleles, they have the darker skin. The recessive phenotype, little a over little a, although these authors did not stick in the slashes that we have come to love and expect. You get the very light skin. Okay, so the F1s are all heterozygotes, they have an intermediate skin color. So this is one gene controlling the trait. If you're homozygous big A, you get one version of the phenotype. If you're heterozygous, there's a second version. If you're homozygous little a, you're a third. What this basically means is you can predict the genotypes based on the phenotypes. There's a perfect correlation between genotype and phenotype. Dark color, we know it's homozygous big A over big A. Intermediate, heterozygous. So here's how genetic mapping works. For a simple trait means it's simple to map, a single gene controls it. So one gene controls this trait. That is, it acts in a Mendelian fashion. So here I've sampled, of course in theory, 8 different individuals, and I'm showing you their phenotypes up here. And I've genotyped all 8 of these individuals at 5 different genes, A, B, C, well, not A, B, C, D, E, F, that are arranged in this order along chromosomes. So you can think of each of these as a pair of chromosomes. So you see their genotypes on each chromosome of each individual. It'll take about 2 minutes see if you can find the genotype. So what gene is it? We want to know which of these genes, B, C, D, E, or F, controls the trait of skin color in this case. So feel free to talk to a neighbor about this, figure it out, and we'll come back together in 2 minutes. Should be fast. Which genotypes correlate with the phenotypes? Okay, so where are your discussions at in terms of figuring this out? Which gene would you like to nominate as being controlling this trait in this example? So I heard F and I heard F. I heard a lot of Fs. Oh, dear. So why do you think it's F? What did you look for to determine that F is the gene that controls this trait?
The darker ones have the homozygous big Fs.
Okay, so every darkest individual has that genotype, homozygous big F. And then what about all the intermediates? They're all heterozygous. And then all of the individuals that have the lightest skin color have a third genotype. So this is perfect correlation. This doesn't prove anything about causation. This is genetic mapping. This suggests a hypothesis. Maybe gene F controls this trait because there's a perfect correlation. Every time we see one genotype at that gene, we see a predictable phenotype. That's how genetic mapping works. You collect phenotypes of a bunch of individuals, you look at their different traits, how they vary from individual to individual. You collect their genotypes at genes across the entire genome, lots of genes, every chromosome. And then you just, nowadays, use a computer program to look for correlations between what the genotypes are of the individuals and what their phenotypes are. Just like you did. This is how it works for a simple trait, where we expect one gene to control the trait. So now I'd like you to try to stump me on a question. Don't make it too difficult. So that example we just talked about involved blending inheritance, right? Incomplete dominance, where the heterozygote has an intermediate phenotype between the two parents. What I'd like you to do is to work on a different situation, just simple dominance. So I'd like each of you, or in small groups if you want, to just invent a phenotype. Come up with some sort of phenotype. Blonde hair, dark hair, happy, angry, can shoot spider webs out of his hand or not, that sort of thing. So make up a phenotype that's binary. You either have it or you don't. And then come up with a gene. Just pick a letter. So down here I've suggested genes L, M, and N, just for consistency. So choose a phenotype, binary, there's 2 different versions of it. Assign that, invent 8 different diploid individuals. So you're going to show me 8 different individuals. Each of them you will have assigned one of 2 versions of that trait. And then I want you to invent genotypes at 3 different loci for each of those 8 individuals. So basically what I'm asking you to do is do the exact same thing I just did with the skin color example. Make up a mapping example. Have one of those 3 genes, 3 loci, L, M, or N, be the one that controls the trait. And have the other 2 be unrelated. So we'll take about maybe 8 minutes, 10 minutes to do this, depending on how quickly things work. Please do raise your hand. I'll come around and answer questions as you go. But when we're done, please attach this to the Google Classroom assignment I sent out at the beginning of class, and then I'll try to work through some of your examples, and we'll see if you stump me. Okay, so what are we going to do to figure out which of these genes, is it L, M, or N, that controls your disposition towards genetics? So what's the first step? So we're looking for a perfect correlation, hopefully. It's usually, in real genetics, it's never perfect, but we're hopefully looking for a perfect correlation between the phenotype and the genotype at one of the loci. Now, it would be helpful if we knew which of the traits was dominant, but we can probably figure it out anyway. What's true about every individual that's frowny face? Is there something common about their genotypes? Right, so they both have little n over little n. Okay, so that's the first thing to do. Take every individual that has the same phenotype, look at all their genotypes, find one thing that's in common. What's the next step? What should we do at this point? There are many ways to go about this analysis. So what would you like to do next? You want to go to the other phenotype and see if that's consistent too? So what's true about all the happy individuals? What's similar about all of them? At any one of the three genes, L, M, or N. We're not sure yet if N is really the gene that controls this trait. Is there anything consistent about their genotypes? Wow. That's impressive, dueling phones. So is big N or little n dominant? Big N seems like it's dominant, which it should be. So every time one of these individuals gets a big N or 2, they're happy. And when you have no big N's, when you're recessive little n, then you're unhappy. Who wrote this one? Did we get it right? Does anyone want to claim it? Or did I get it wrong, and so you're not going to tell me anymore? Who did this? This was good. Hit the spot. No? Okay. It's fine. I respect anonymity. So what about this one? Curly hair and straight hair. So we're looking for something that's consistent with, I don't know, let's say straight hair. What's the same about all the individuals that have straight hair, genetically? For some reason, it's always the gene on the bottom that controls the trait, which is not true in genetics. So everybody that's got straight hair seems to have homozygosity for little n. The thing to check, to double check your work when you do these sorts of simple genetic mapping experiments, is you want to ask, are there individuals that have 2 little m's that don't have straight hair? That's a step we didn't take on the example previously. But you really want to look for the best correlation between genotypes and phenotypes. So in this case, again, it's true, it seems, that every individual that's got curly hair has at least 1 or 2 capital M's. Did we do right? Who wrote this one? Yes? Did we get it? M controls the trait? All right. And capital M was dominant for curly hair. Okay. Outstanding. So that's how you do simple genetic mapping. Does the genotype match the phenotype? Does the phenotype match the genotype? Any questions about that? So that's exactly like questions that might show up on midterms, third exam. That's easy compared to complex traits. That's why they're called simple traits. They're simple to map. They're controlled by one gene. Then we get things that geneticists are more and more interested in, like complex traits, like human height. This is a famous picture that was taken, as you can probably tell, if you can see what they're wearing, a long time ago. And we could do the same thing if you're interested. Let me know. We could do this next class. You just categorize students by height, and this is basically a histogram. How many students in the class were different heights? So we've got, what is that, 4 students that are 5'0", 4 that are 5'1", 6 that are 5'2", and so forth. So you see that there's a distribution of phenotypes. It's not binary. It's not curly hair, straight hair, It's not widow's peak, no widow's peak, attached earlobe, unattached earlobe. It's a complex trait. And it's complex because this is a trait that's controlled by multiple genes, not just a single gene. There's not one gene that tells us how tall one of us is going to be. In fact, it goes beyond genetics even, you might appreciate. It's environment too. Your height has partly to do with your genetics and partly with your nutrition, where on Earth you live. Are you higher up? Are you closer to the center of the Earth, gravity? You heard that the astronaut station commander Scott Kelly returned to Earth after a year in space a few days ago? He was in space for a year. He came back and he was, what, 2 inches taller? Because his spine decompressed that much over the course of time, he was weightless. And that's why each of us is taller when you get up in the morning than you are when you go to bed. Because we spend all day, our spine is being compressed. You wake up in the morning. So if you ever want to grow, go to the doctor and have your doctor's appointment early in the morning. You're taller. Ride all the good, no, you guys don't have to worry about how tall you are on rides at Disneyland. But, right, so this is a complex trait. There are lots of factors that control human height. So how would we ever go about figuring out where are all of those genes that control this trait? Back to our skin color example. Skin color in most species, including humans, is not a simple trait. Like it was portrayed earlier, it's really a complex trait. There are lots of genes that control how much pigment gets put into our skin. So what the matrix looks like is more like this. Assume that there are 3 different genes, A, B, and C, that control production of pigment in skin cells. So if you have 2 little a's and 2 little b's and 2 little c's, you have very light skin. Or you could have one of those 6 total alleles that produces more pigment. You have slightly darker skin. You could have 2 alleles at any one of those 3 genes that would give you slightly darker skin, like here. And if you inherit all 6 dark alleles, then you have the darkest possible skin type. In complex traits, you don't get binary traits. It's not curly hair versus straight hair. It's not tall versus short. It's not happy versus sad. It's a trait that's continuous. It forms, as shown here, a continuous distribution. So there are not discrete traits. They're varying traits, like height. Right? Height isn't binary. It's not you're tall or you're short. It's how tall are you? We can measure it. And this is how, very simply put, and this is definitely something that scientists use computers to do, not doing by hand, although a grad student and I have been doing this by hand in the lab recently, and it's complex, which is why it's called complex trait. So what we want to do in this case is we've got 2 versions of a plant, one that has few hairs that are produced on the leaves, and one strain of this plant that produces many hairs. So you've got the red genotype here that produces few hairs. Blue, you get many hairs. So we know there's a genetic difference between the blue strain of the plant and the red strain. And we want to figure out where are the genes that control this trait. So you make an F1. It's got an intermediate trait. It's kind of hairy, not super hairy. It's intermediate between the parents. And then you sibling mate, or you self, that F1 generation many times, in this example, until the 8th generation. So you start from F1, you mate siblings with each other, you keep mating siblings together for 7 generations. And you get a panel of chromosomes down here, like is shown at the bottom, where every individual plant has a slightly different genotype. It's got different patches of the chromosome where they have red alleles. Like this chromosome on the left is completely red. But then we have chromosomes that are part red, part blue. So what we need to do is figure out what part of this chromosome, where is there a gene located top to bottom along each of these chromosomes that controls this trait, hairy versus non-hairy, degree of hairiness of the leaves. We start exactly like simple trait mapping. Can you find, instead of using letters, we're using colors. Can you find a region of the chromosome where the color at one part of the chromosome has some sort of correlation with whether or not that plant produces hairy or non-hairy leaves.